Now Martin Tobin is going to talk about UK BiLEVE, which was really a piece of prescience that made us start to realise that it might make sense to do genotyping on the scale of UK Biobank with this project, UK BiLEVE, which was the first genotyping project within UK Biobank. Martin.
Thank you very much for inviting me to speak. And so, are we ready with the? Okay. So the UK BiLEVE study, as Rory's just introduced, is a study of lung function and of extremes of lung function. And part of the motivation for studying these traits are to understand the genetic determinants of chronic obstructive pulmonary disease, or COPD. And as mentioned, this was the first genetic study in UK Biobank. And this was actually initiated before we actually had DNA extracted from the stored blood samples, or at least processed blood samples in UK Biobank. So we actually began by analysing the lung function test data on all 500,000 UK Biobank participants. So tremendously rich data set. There were actually data points recorded every 100th of a second during the whole of the spirometry blow curves that were available. So this gave us a richness of data to be able to undertake very detailed quality control of these data. And for this particular study, we decided to use really strict criteria for controlling the quality of these spirometry curves, and that was partly because we were sampling from the extremes. So as you can see on the slide here, we had a sampling frame of just over 275,000 individuals whose spirometry met these very strict quality criteria, and these are defined by the American Thoracic Society and European Respiratory Society. So within that very large sampling frame, we studied never smokers and we studied heavy smokers. And within each of those groups, we sampled from the extremes of the lung function distribution and from the middle of that distribution. And the particular measure that we used is one called FEV1, which is the forced expiratory volume in 1 second. And that was corrected for age and sex and height. So that enabled us to compare not only the extremes of the lung function distribution, but also to compare never smokers and heavy smokers. And that study enabled us to discover a number of novel variants that hadn't yet been reported as being associated with smoking behaviour. And we were also able to discover novel genetic determinants of lung function and of chronic obstructive pulmonary disease. So that was reported in a journal called The Lancet Respiratory Medicine in September last year. And simultaneously, we were working via a big consortium of cohort studies called the Spirometer Consortium, which additionally reported some further variants associated with lung function last year as well. So since that time, we've been able to bring these resources together, and we've been able to study genome-wide association studies. Genome-wide association, it's abbreviated as GWAS here, across all of the UK BiLEVE participants, so that's just under 50,000 individuals. And we've also been able to follow up the most promising signals for those quantitative lung function traits in a further 95,000 individuals, and that's bringing together additional individuals in UK Biobank that have been genotyped since our original study, together with populations from the Spirometer Consortium. So the 3 lung function traits that we've studied are FEV1, the ratio of FEV1 over FVC, and that ratio is often used as a diagnostic criterion for diagnosing chronic obstructive pulmonary disease, whereas FEV1 is used in order to define severity of disease. Forced vital capacity we also studied. So This approach has enabled us to discover 43 new signals associated with lung function. And this now brings to 97 the number of independent signals for lung function. And these signals have given us some important clues. For example, they point to a number of proteins for which either approved drugs exist or for which drugs are in development. So this can give us some important information which might help in terms of targeting future treatments. We've also had some important questions as to how important these 97 variants might be in terms of susceptibility to this disease that we call COPD. So we've actually followed up these variants, or at least the vast majority of them. 95 of them were adequately measured in other studies. And we've been following these up in 10 additional European ancestry populations that have collected COPD cases and controls. And we've also been studying them in studies of other ancestries. So this includes the China UK Biobank. Amongst those case control studies for COPD, in particular those studies that have some of the most detailed phenotyping on which to base the diagnosis of COPD, so that includes what's called post-bronchodilator spirometry, we're actually finding fairly similar effect sizes for our risk scores as we are in UK Biobank. So we've undertaken some further risk modelling in UK Biobank. And if we categorise our UK Biobank participants according to the number of risk variants they carry across these 95 variants, so we break them down into 10 equal-sized groups that we're referring to as deciles here. And we can see that individuals in the lowest decile are individuals in the highest decile are at more than three times higher risk of developing COPD than individuals in the lowest risk decile. So we are now demonstrating the relevance of these variants not only to quantitative lung function but also to COPD. So I'd just like to thank the participants of UK Biobank, without which, of course, this wouldn't be possible, but also the vast team of scientists who've made UK Biobank possible, who've kept the wheels turning with UK Biobank, and who've been so collaborative. I'd like to thank all the follow-up studies who've enabled us to actually demonstrate the relevance of these findings to COPD. I'd like to thank the Spirometer Consortium members, all the UK BiLEVE Consortium members, Affymetrix, who've been very good to work with in terms of the array design and the genotyping. And I'd like to particularly thank my co-PI, Ian Hall, and co-investigators, Louise Wayne and David Strachan. And David's in the audience. And I'd particularly also like to thank Nick Shrine, Maria Solar-Artigas and Vicky Jackson, who've undertaken so much of the hands-on analysis in our team. Thank you.